A well-designed drug delivery system is as important as the pharmacological activity of a drug, since the therapeutic efficacy of many drugs is often limited in the administration by their bioavailability, solubility, stability, and safety. After administration of a drug, only a small fraction of the dose actually arrives at the target receptors or sites of action, and usually most of the dose is wasted either by being taken up by other tissues or by decomposition before arrival. Drug delivery system is used to administer a pharmaceutical compound to achieve an optimal therapeutic effect. This technology may involve control of drug release rate, improvement of therapeutic index, minimization of drug degradation, and reduction of drug toxicity in the body, offering means of optimizing therapy with established drugs. In recent years, drug delivery is becoming an increasingly demanding science due to the substantial decline in the rate of appearance of new drugs. As the most versatile class of materials, polymers have gained a worldwide interest. The large number of types and structures of polymers can basically be synthesized, offering advanced functions. Biodegradable nanoparticles is one kind of polymers frequently used to improve the therapeutic value of medicinal drugs and bioactive molecules. Many nanomedicines of the dreadful diseases like cancer, AIDS, diabetes are in clinical trial phases, and some of them are commercialized. Me and Lan are going to talk about polymers for drug delivery. As a new generation of the polymer-based colloidal carriers, polymers have attracted rapidly growing interest. We can see from the structure attached. Polymers are artificial vesicles that contain an aqueous solution in the core, surrounded by a bilayer membrane. The bilayer membrane is composed of hydrated hydrophilic coronas, both at the inside and outside of the hydrophobic middle part of the membrane. The aqueous core can be utilized for the encapsulation of therapeutic hydrophilic molecules, and the membrane can integrate hydrophobic drugs within its hydrophobic part. Due to the relatively thick membranes, polymers can be rather stable. Next, we will discuss how to prepare polymers. From the graph, we know that many techniques can be used to prepare polymers by self-assembly of amphiphilic block copolymers. What is block copolymer? It comprises two or more homopolymer blocks. Each block is polymerized with a specific monomer or a combination of monomers that have unique physical-chemical properties in the polymers. Block copolymers are used for creating self-constructing polymers with a variety of properties and potential applications. Polymers with a range of properties can be produced by applying block copolymers with different molecular weights, functionalities, compositions, and molecular architectures. The most important preparation methods are generally classified into two groups: solvent switching techniques and polymer rehydration techniques. First, we will talk about the solvent switch techniques. Polymers are formed by first dissolving block copolymers in an organic solvent, which is a good solvent for all the blocks present, followed by hydration of the solution. This procedure renders the hydrophobic blocks insoluble, triggering copolymer self-assembly into polymers as a result of increasing interfacial tension between the hydrophobic blocks and water. Therefore, this technique is also called as phase inversion. Then, we want to know polymer rehydration techniques. Polymer rehydration techniques are based on the hydration of amphiphilic block copolymer films to induce self-assembly. Polymers are first dissolved in an organic solvent, and then a thin film is prepared by evaporation of the organic solvent. Subsequently, the films is hydrated by the addition of water. Next, we will discuss how to characterize polymers. The most common tool to investigate polymers in an aqueous dispersion is dynamic static light scattering. This technique has been mainly used to determine vesicle size and size distribution, but also for determining the critical aggregation concentration, vesicle disruption, and change in the size with variation of pH or temperature. Typically, polymer rehydration produces polymers with a broad size distribution, and therefore, the polymers obtained are subsequently sized by sequential extrusion through filters with different pore sizes using a high temperature. Nanocarriers have been explored for the delivery of therapeutic and diagnostic agents. However, a number of questions of whether the properties of carriers are suitable for biomedical applications are still remaining, caused by the interactions of polymers with proteins and cells. One of the main problems encountered with the application of nanocarriers is the rapid clearance of carriers by nanonuclear phagocytes in vivo. Nanonuclear phagocytes is a part of the immune system that consists of phagocytic cells located in reticular connective tissue. It is generally known that the clearance of nuclear carriers starts with adhesion of proteins. The carrier protein complex can be bounded to appropriate receptors on the phagocytes, allowing subsequent adhesion of the complex to the phagocytic cells, and eventually internalization of the carriers.
Conventional treatment of many diseases involves the direct use of the needed drugs, which leads to uncontrolled or undesired drug release time and locations in the body. Drug delivery polymers are meant to improve the pharmacokinetics of the drug in the body by controlling the drug's concentration in the bloodstream over time, targeting capabilities, absorption, and avoiding immune responses. Other positives include possible financial benefits and improved patient compliance. To achieve the best possible drug delivery system, it must be able to incorporate specific responses to physiological environments. A common structure of drug delivery polymers are shown. A vesicle of the designed polymer with a hydrophobic end encapsulates the treating agent in the core. The other ends of the polymer on the surface of the vesicle contain reactive groups or ligands which are used for specific targeting in the body. As for the benefits of polymers, unlike other materials used for drug delivery, polymers offer increased biocompatibility by reducing immune responses and improving stability in the bloodstream and solubility. For example, poly-2-oxazolenes has stealth behavior in the body, meaning it will not interact with other particles in the bloodstream. This can be used as an alternative to PEG, which is commonly used in medicine, when a patient exhibits an immune response to PEG. Additionally, polymers are cheap compared to other materials. I'll now go over multiple methods in which the polymers are used for a controlled drug delivery system. Conventionally, a dosage of drugs results in a burst of its concentration in the blood and then a slow decrease in time. For chronic treatments, multiple doses are administered in intervals related to the half-life of the drug in the blood. The main types of release methods are the following: time-controlled or diffusion-controlled, and also smart or reactive release systems. The first method are extended release formulations. These release a dose at a sufficient amount and then constantly release the agent over time at an adequate rate. There are two types: a polymeric matrix, which controls the diffusion rate through the polymer network. These are usually hydrogels or nano to micro particles. This type obeys Fick's second law by treating the problem as a pseudo-steady state. The other type is a polymer-coated capsule in which the permeation determines the release rate. An example of the first type are Capinol tablets. They provide diffusion-controlled release of morphine sulfate. And an example of the second type is Cenemit CR, which controls the release of carbidopa and levodopa by eroding the polymer's coating. Another type is sustained release method. These can follow zero-order kinetics, which slowly release the agent over a long period of time. Osmotic pump systems work this way, and the rate is affected by the solubility, size of pump, and osmotic pressure. An example is the Adalat OROS product. It works as an osmotic pump releasing nifedipine Lasers are used to drill holes on the tablet's water-permeable coating. Then the osmotic pressure as the tablet passes the body controls the release of the drug. The next type is the pulsatile release method. These are released after some lag time or in a pulsatile fashion as shown in the graph. It comprises a sensor and a delivery system. The benefits are that they release the agent at the proper time or location automatically. An example is the following. For diabetes, a drug delivery polymer would cause a pulsatile release when blood sugar levels rise. Glucose oxidase converts glucose to gluconic acid. The decrease in pH causes insulin to release due to the degradation of pH-sensitive polymers. The previous examples are considered to be time-controlled release methods. The next methods are smarter systems which work due to changes in the environment. The first are redox-based methods. Polymers containing easily altered linkages are an option for responsive biodegradation. Some examples of these focus on linkages that are acid-labile, like polyanhydrides, polylactic or glycolic acid, and polybeta amino esters. Disulfide linkages are also used since they are unstable in reductive environments, where they are cleaved for favorable thiol groups. Next are temperature and pH-based methods. For pH-based methods, these are especially relevant in the GI tract and also to other areas such as dosing diseased versus healthy tissue, which do vary in pH. This is done by hydrophobic transitions based on pH. These polymers are tuned by adding hydrophobic functional groups in the core and by changing the cross-linking density. When the polymer is exposed to a specific environment, it degrades and the drug is released. An example of this is in tumor cells, where the pH is lower than healthy tissues. As for temperature-based methods, the lower critical solution temperature of a polymer is where below it the polymer is a coil and above it it is a globule. This is due to the hydrophilic and hydrophobic balance in the polymer. This varies with the temperature, as the polymer-polymer hydrophobic interactions dominate at a temperature above the LCST, and the opposite when below the LCST. The temperature range can be shifted. This is done by adding hydrophilic and hydrophobic functional groups. So, for controlled drug delivery, it is useful to shift the critical temperature phase transition to a desired temperature range. I'll now talk about the feedback-controlled release method. The concept of the feedback-regulated drug delivery system is a future prospect for drug delivery polymers. Research effort has been placed to design nanoparticles that are capable of feedback-controlled drug delivery. These types of systems take advantage of a biochemical triggering agent. Then, the drug release rate is triggered by the triggering agent's concentration. It would be very useful if a disease would release some kind of signal as a triggering agent. Then, the magnitude of this signal would cause a release of an appropriate amount of treating agent from the nanoparticle. As we can see, there are many methods in which a controlled drug delivery system can be prepared. The specifics of a disease is where the information on how to tune a polymer for either timed release or location-controlled release is obtained. This leads to higher effectiveness of the drugs, their administration frequency, and it helps with improved patient compliance. Finally, it is important to note that since controlled release systems cost much more, their use must be justified.
Here are resources found. Thanks for watching.